Good afternoon, Mehershad. How are you? Mehershad, can you hear me?
Hi, ma'am. Sorry, I was on mute. But
Yeah,
yeah, I can hear you.
you. Nice to see you here.
Great to see you again, ma'am.
How is everything on your side?
Everything's good. I just came to lab earlier today because I'm going to Melbourne on Monday, Tuesday, Wednesday. So I need
Oh,
to finish some work before that. So everything's good.
yeah. Nice to see you after so
Yeah.
long.
Yes, I
Yeah.
actually I came to meet everyone, but I think that day there were examinations and stuff. So I think you might have left by the time I actually came.
Okay. Then I missed you.
Yeah. But again, I think it'll be tricky now because I heard about your farewell, but hopefully we'll still catch up.
Yeah. Sure. Why not? So you are now in Melbourne.
No. Sorry, I'm in Brisbane, but I'm going to Melbourne for the first time
But
this
you are
Monday.
going to Melbourne. Okay.
Yes.
Nice. We are eager
Yeah.
to hear you. Yeah.
Yeah.
I read some like a few points of your Pluto Gets Vaccine. I think it should be very wonderful book, right?
Thank you so much. Thank you.
I'll order it. It's very
Yes,
nice.
thank you.
So you are going to talk about that also today?
A little bit. Mostly, I'll focus on
Yes.
the
Yeah.
science opportunities, but I'll touch upon it a little bit just in
Yeah.
case people are interested.
Yeah. Sure. Do it. Like it's very nice. Like nice way of putting it. I just read some reviews. That's it.
Yeah, I mean, it's very oversimplified, but it is done in a way that will help people understand, especially children.
Yeah,
But yeah,
I
from an
know. Yeah.
immunology point of view, it's very oversimplified.
Yeah. So, how is everyone? Ria, Taiba, all good?
Sorry.
No, I'm asking other students also, like
Oh,
how they
okay.
are. So, it's night for you there?
It's actually just sunset. The sun is going to set
Okay.
in I think 10 minutes. Yeah, so late
Okay.
evening. Yeah.
Okay, late evening. Yeah. Must be summer now for you.
Yeah. It's actually really
Summer.
right now it's
Yeah.
really hot. It's just like Mumbai summer
Very.
right now. Right now it's a 35, 36
Okay.
peak summer. Yeah.
Okay. Yeah. Nice. Hello, am I audible?
Yeah, I can hear you.
Okay.
Hello. We will be starting in just 2 minutes. So, hello. Am I audible?
Hi. Sorry.
Is the screen share visible?
Yes, yes. Have I done it correctly?
Hello. Am I audible?
Yes, yes.
Hi, sir. I would just like to request you to close your screen sharing because I would like to present.
Sure.
Thank you, sir.
Is this fine? Yeah.
very very awesome to all. Yeah. I will call you the second question of humans of sciences, our electric packaging. The question of Indian standard 2023. I will demand the one. Who is the director of scientific eradication? Yeah, it was very tomato as an instrument. Yeah. One out the JHC like indicator. But this year, unlike initiative, immense pleasure to introduce our speaker for the day, our classmate, friend, Mr. Mehershad. Wadia. Working at also assistant at England Bay. He working on the project. He has project and work experience in different field of sciences, which you can see here on the screen. He has leadership experience and community work. He was awarded as Brisbane International Student Ambassador and also as University of Queensland International Student Ambassador. He has the award and scholarships to his name. We can see here the different awards and scholarship he has to his name. So, Mehershad,
Hi. Yes, it's sort of, it's a bit robotic, so I can't catch everything. But I think maybe, I don't know if it's my internet or on your end, but I can sort of make up what's happening.
Anyway, so as I mentioned, we are really proud to introduce our classmates who was a resource person, achieving all these beautiful things, achieving life. We went through the dedication of this project's publication. Now we going to that very the award they shot war in UK as Best International Film Award in Australia. He's been a writer who spent the office in International Film Festival as well. So this is the very formal introduction that we've given with respect to Mehershad's biodata so far, and now we'll probably come to the
Oh
not very
no.
hold on. So we've seen so this is then trying to see that. Okay, this was the explanation. Please go on. Please don't look at me in some manner. So, yes. Check it, again, check it, check it. It was asked like okay, so this was like levels. You had now he knows that that's here to give you so thank you so much, ma'am, for the lovely introduction. So I hope you liked it. Hear me? Hello.
Hi. Sorry, I can't. I can't hear. It's all robotics. I can't really hear. But I'm trying to learn how to read lips. So I think I got most of it.
Did you see most of it? Did you see the PPT?
I can see most of the images. Unfortunately, I saw most of the images, and I'm sorry you had to see them too. So I saw the images.
Like, okay. Thank you so much, ma'am, again. So, I would request you to start this session.
Sure. Can everyone hear me? I'm sorry, I'm not sure what's happening with the audio. I can't really hear anyone, but as long as you can hear me, that's more than enough. So can everyone hear me?
Oh, we can hear you.
Okay, perfect. Okay. It's really hard to start a presentation and to be in any authority to give advice to anyone after seeing those images and after hearing about some of the earlier days in life sciences. But I'll still try. I just realized because of this share screen, you can't really see my mouse pointer. But so just try to pay attention on what I'm saying. A lot of the things on the slides will not make sense until I mention it. Because this is Department of Life Sciences and because we're scientists or people who aspire to be scientists, this will be a very scientific presentation. If at any point you have questions, please stop me. If at any point you can't hear me or can't see the screen, please stop me. Right. So before we start, there are some experimental considerations that we need to make before taking up any experiment. In this case, the case of the talk, it's important that you have scientific curiosity. So please stop me, please ask me questions at any point. However, time-sensitive assays exist in science, which means that if you have questions that might only be relevant to you, please be considerate of other people's time. So please feel free to engage me later after the talk. I'll be happy to stick around. But if you have questions that might be relevant to most of you, please stop me. Please ask any questions. Right, low sample size, which means that anything I say it because it's worked for me. But n is equal to 1. It's just worked for me. So maybe what I say might not work for you. So take it with a grain of salt. Terms and conditions applied. And also, there's no control group. Maybe all the things that I will suggest to you, maybe if I had done things differently, maybe I would have had a Nobel Prize by now. We don't have a control group. So again, I'll tell you things that have worked for me or things that I think I would do things. If I could do things differently, this is how I would do things differently. And I hope it helps you. But just understand that life is complex and there are a lot of moving variables as is in science. And also there's natural selection, which means that the fact that you're in Department of Life Sciences and the fact that you're here attending this talk means that we've already selected people who will have great careers by the virtue of you being here and by the virtue of you being in life sciences. So already you're naturally selected for having good careers. So congratulations on that. And I don't know if you can read this, but acknowledgements and references. It's very strange to give this talk because any advice that I've ever gotten or the advices that I've listened to, they've come from the people who are in life sciences. They're the teachers and the professors at life sciences. So it sort of feels like for some reason there's this middleman where I'm supposed to tell you things that they can tell you because they're the people that taught me most of the things that I know. So acknowledgement to them. So these are the experimental considerations that I want you to keep in mind while we go on the scientific quest. Right, incubation. I think most of this is already covered by Sakina and Zinia. I did my 11th and 12th at Jahind. Also did my bachelor's of life sciences in Jahind. And then I went to the University of Queensland for my master's. And yeah, unfortunately, I don't have a PhD by now, but yeah, I should have had it a long time ago, according to my mom, but maybe I'll have it soon. So let's see. Now let's move on to background literature. a lot of this is already mentioned. It's just some of the projects that I did. If you're interested in these topics, I'm happy to speak about my projects at length separately. But I don't think I'll bore you right now, especially because it's a Saturday. But essentially, I started off during my master's. I worked on two plant genetics projects, and then I felt that I wanted to work on something that is more closer in the pipeline to human research. So I've sort of shifted from plant genetics to genetics in C. elegans, which is a model organism. So it sort of feels like I'm moving in this unilateral direction from plants to now like a worm. Maybe then I'll go to mice. Maybe then I'll go to humans. So maybe that is my trajectory. So currently I'm working at the Queensland Brain Institute. That is where I am right now. And I mean that literally and metaphorically. I am currently also geographically at the Queensland Brain Institute. And the two projects that I'm working on are the role of UNC71 in axonal regeneration. So C. elegans, the nematode, has this beautiful quality of being able to regrow their neurons, which, as you can imagine, would be very useful for us to do if we crack the science behind it. And I'm also looking at, so there is this novel gene that we found in the lab a few years ago called OSP1. We call it OSP1, OSP1, because it's supposed to be, it's supposed to stand for oxidative stress protection 1. So as the name suggests, we're trying to understand how it protects neurons, not our neurons necessarily, but C. elegans neurons from oxidative stress. And hopefully this sort of teaches us things about the machinery that we can use with humans. And one of the very common questions that you might probably have is Mehershad, hey, you're looking at nematodes or worms, especially microscopic worms. So how is this research ever going to be applicable in humans? And that's a very fair question. But the reason is because of evolutionary conservation, where most of the cellular processes at the very microscopic level sort of evolve independently, but they have a lot of parallels. And at some point, humans and C. elegans, the worm, had a common ancestor. So there's actually a lot of things that these small worms teach us about us, especially because we have billions of neurons, and it's hard to really understand what's happening there. So yeah, moving on. So this is just background literature, some of the stuff that I've done. Now moving on to alternate mechanisms. With most pathways that I've studied, there are always
Hello.
redundancies. Yeah.
Sir, I'm sorry to cut you, but actually we are having a little technical problem, so we are not able to hear you. So we are just sorting that issue. Can you please wait for 5
Oh
minutes?
yeah, for sure.
I'm so sorry, sir. Thank you. Hello.
Yeah. Can you hear me now? Is this better?
No, sir, 2 minutes.
Sure.
Sir, can you say something?
Yeah. Is this helpful?
We are actually not able to hear you.
Should I try disconnecting my earphones and try with another? Sorry?
Yeah, you're audible now.
Okay. Feel free to stop me if I'm not at any point.
Yeah. We're actually just seeing that if sir, you can start with the
Do you know at what point, till what point was I audible? And if I wasn't audible at all, that's fine as well.
Mehershad sir, you can go on.
Yeah. Do you know at what point did I stop being audible?
What? Yeah, start the slide again, please.
Sure.
From the start, from your previous
Okay,
slide.
okay.
Yeah, thank you.
This one?
Yes, you can start.
Yeah, thank you. So yeah, a lot of these things have already been covered by Sakina and Zinia. So I won't. I mean, it's pretty straightforward as well. I'm very proud of my PhD joke there. So this is just incubation. This is education and stuff. Now let's move on to background literature. So again, a lot of these things have been mentioned previously by Sakina and Zinia. But if any of this looks interesting to you, feel free to. So I'll just resize. There's a Zoom overlay that's come. Okay, cool. If any of this looks interesting to you, feel free to engage with me later. I'm happy to talk about my projects. But if I start now, it'll go on for an hour. As should most. As should most scientific conferences. So as you can see earlier, during my master's, I got into a lab that focused on plant genetics. And later on, I felt that perhaps I'm a bit more interested in things that are clinically relevant or are more relevant in humans. So I switched to the lab where I am now, which is the Queensland Brain Institute. And we essentially work on a microscopic nematode called C. elegans. C. elegans is actually, it's normally mentioned alongside Drosophila, the fruit fly. You might have heard it either as a fill in the blank in a textbook, because it's one of those textbook model organism example that we come across. So C. elegans is a round, tiny, microscopic worm, but it's transparent, which is really helpful because it's transparent, we can see its neurons while they're in them. So we can study the neurons without having to dissect or do anything. We can study the neurons while they continue to live their life, which is extremely insightful. So the two projects I'm working on, first of all, is looking at UNC71. This is a gene that encodes an ADAM enzyme. I won't go into what an ADAM protein is, but we're essentially trying to see how this gene or protein helps in axonal regeneration. C. elegans also has this beautiful quality of being able to regrow its neurons. So as you can imagine, it would be extremely helpful for us to understand how we could possibly regrow our neurons so that we can get rid of a lot of these neurodegenerative diseases like Alzheimer's and motor neuron disease, etc. And the other project that I'm involved in is concerning OSP1 or OSP1. So this is a novel gene that we actually found in our lab a few years ago. And OSP1 is the name that we've given it because it stands for Oxidative Stress Protection 1. And as the name suggests, it helps protect neurons against oxidative stress. So again, I won't go too much in detail about these very specific projects because they might not be relevant to you. But if you're interested or if you're interested in the field, please feel free to ask me questions later or connect with me. So these are just some background projects that I've been involved in. The other thing I wanted to mention is that as Zinia and Sakina already mentioned, my life has been sort of a dichotomy between science and creative projects. So I just wanted to mention that other than these things, I'm also interested in filmmaking. I'm interested in drama. I'm interested in writing and comedy. And recently, one of the things that I'm really proud of is I recently wrote a book called Pluto Gets a Vaccine. It's a children's book. It's very trivial. Especially for adults, it's very basic. But I really like it because this, and I'll cover this later, but this sort of shows to me that if you're interested in science, you just don't have to be a researcher that works on a lab bench. There are a lot of things within science that you can do. Especially now that science has, unfortunately, but after COVID, science has come in sort of the popular culture. A lot of people are interested in vaccines and viruses and stuff like that. So there are a lot of things that are opening up. So these are just some of the things that I've done so far. And now I'll move on to, since this talk is a research experiment, let's come to methodology. These are the things that I've done. Now, what made me think that I want to do them, or what were my thought processes behind going to University of Queensland, for example. So that probably you could also have a similar thought process, so you can learn from the things that I learned in making whatever decisions you need to make for yourself and for your career. Why and how I chose the University of Queensland. I was really interested in research. One of the reasons that I chose to go to a university that's outside of India is because I felt that there was a little bit more focus on research, and research that is that has more of an impact, that is done more commonly, that happens more frequently. Which is why I chose the University of Queensland. It's really known for its research. There are a lot of institutions within the University of Queensland, one of them being my current employer, which is the Queensland Brain Institute. So the reason, the way I went about it is there are metrics, and there are always rankings and things like that. Different people have different organizations have different rankings of universities. A lot of them are also ranked by focus on research. So you can go through those metrics, see what universities cater to the field that you're interested in, and to if it has sort of the strength in faculty, if it has a lot of researchers that it employs, because that just means that you have a lot of options to choose from. The reason I mentioned field is as you can see, I was into plant genetics, now I'm into neurobiology. So I necessarily, life sciences was great for me because I'm very holistic in my approach with science. There's not a particular niche that I like to limit myself to, or I'm necessarily interested in a very small niche within the field. So life sciences was great, and University of Queensland was great. But the reason I mentioned field is because, for example, I know the University of Queensland is one of the only few universities that has a strong focus on physiotherapy and exercise. There are masters that you can do in very particular aspects of exercise and things like that. So a lot of universities have their own specializations, own niches that they're really good at, that they're ranked very well at. So if you have a particular niche, you want to go to a university that caters to your need. So then the other thing is, in Australia at least, and I'll speak about Australia, but in the US, there's something called the Ivy League colleges. But I'll stick to Australia because that's what I know more about. In Australia, we have the group of 8 universities, which is essentially these big sandstone universities that were made hundreds of years ago. That's the UNSW, the University of Queensland, University of Western Australia, etc., etc. So these are these 8 top universities in Australia. But there are also a lot of other universities, a lot of new universities. You might have heard of the Deakin, RMIT, etc., etc. And there's always a debate that international students suffer from, which is where do you go? For example, in the context of the US, maybe you're thinking, should I go to an Ivy League, or should I go to a smaller university that might not be Ivy League necessarily? And it feels intuitive that you want to go, if you can go to Oxford, go to Oxford. And that seems intuitive, but I don't necessarily agree. Is because a lot of times the smaller, newer universities, first of all, they are more invested in taking care of you. And they have a lot of incentives that they want to use to incentivize you to come to them instead of the bigger universities. So if you're looking, and I'll touch upon this later, but this is one of the things that I thought about, is that if you're looking for universities to go to, one way that you could do that is either go to a really well-known university, but the fees will be higher, scholarships will be scarce. Alternatively, you can go to smaller universities, which have good scholarships and will take more care of you. But obviously, it's important that you do your research there, because some of the smaller universities might actually not take care of you, but some of the smaller universities actually will. So it's important to look at what faculty they have, how well the faculty has been doing, what researchers they house, etc., etc. One of the reasons that I still went to UQ is because, like I mentioned, UQ has so many research institutions that I knew that studying here would be helpful, because I can then do research internships at these institutions. So that's something that you can weigh for yourself. The other question, that if you're at some point looking forward to doing a PhD or a master's or an honors year abroad, you might be thinking whether you want to do, for example, a master's by coursework or master's by research. Different people have different preferences. I'll just quickly tell you the pros and cons that I've noticed with both of them. With coursework, the pro is that it's very easy to get into because it's just the same degree and there's 3 buttons that you click to apply. And it's very straightforward. The con there is that a lot of the coursework classes that I attended were actually worse than the classes that I had in Department of Life Sciences. I remember texting Nilofer ma'am saying that the things that you've taught, they're teaching the same things you are, but worse. Which is why I focused on lab-based courses. I think in coursework, I think you can choose your own courses. So you might prefer to choose lab-based courses. And the reason is that there you're actually doing research. You're not just reading books that you could do here anyway. So if you're going all the way there, I feel like it's beneficial to probably go for lab-based courses if you're doing masters by coursework, for example. Masters by research is hard is because a particular supervisor has to approve you before you can get admission. And you might ask, Mehershad, how will the supervisor approve me before I'm even there? Because you can't get a visa before you get the admission, and you won't get an admission unless the supervisor approves you. For this case, you either need to be very good at networking, or you need to have some connection. Maybe you know the professor, maybe he gave a talk in India, or maybe you know a researcher that knows that professor. Because you need the supervisor to approve your admission and sort of say, yeah, I'll take this person in my lab, and I'll let this person do a master's by research in my lab. But if you can do that, then that's the way to go, because that's basically a mini PhD, and that's very, very useful. Because it's at its core, it's fundamental research experience. You'll be doing your own project, and you'll be in the lab, and I think that's very valuable. Disadvantages that I felt is because, again, I think things are changing now, but I think a lot of the universities and a lot of colleges within India were not as strongly focused on research, on pure research. There's a mix of research and there's a mix of practical lab experience and courses. But it's extremely hard unless you're exceptional or have networks. It's extremely hard for everyone to sort of have an internship at TIFR or ISC, etc., while also doing your degree. I know Jahind has now become autonomous, so maybe it's different now. But back then it was very rigid. You couldn't just go do a research project somewhere as easily as you can do here, which is why I feel like there are some disadvantages that we have with research experience. Someone who's doing a bachelor's here has been in the lab, a proper functional lab, much more than I had, unfortunately. So that is a disadvantage that you should be aware of, but it's not a disadvantage that you can't overcome. And one of the things that are extremely helpful back home and also here is if you can get internships. Thankfully, Life Sciences had internships during the summer vacation. We had internships at a blood bank, a pathology lab, and Relabs, the stem cell company. And if you choose to go abroad, you should definitely make most of your time there by enrolling in summer research projects. A lot of the summer research projects you can actually volunteer, so you don't have to pay for the courses. And the summer research projects that I did, I got selected as a scholar. So you actually get paid to not only do a course but also do research, which is really cool. It doesn't pay a lot, but it's pretty good. So these are just some of the things and considerations that I had when I made any of these decisions. So now moving on from methodology, let's go to future directions. Now you must be wondering that you're in life sciences, you're doing your bachelor's, maybe you're at FY, SY, or TY, but what next? And that's a question that I had. It's a question that I still have. But here's my understanding of it. It's not complete. Also, I could have just Googled opportunities in life sciences, but I didn't. That is another set of research that you can do for yourself. But this is just what I've seen around me, that I've seen with my colleagues, with my friends. So I know that this is firsthand, and I know that this is very much based in reality. Opportunities, obviously, academia is the big one. I think that's the most conventional route. Is that you can get into academia, you can work in a lab, like I'm working at the Queensland Brain Institute, and you can work at different levels. There's sort of a hierarchy, and you can be at any level that seems right for your career. So you can either start as a technician, which means that you don't need a PhD. You don't even need a master's necessarily, but I'll come to that later. Within academia, you can be a technician. Then obviously, if you do a PhD, then there's the postdoc, the assistant professor, professor, group leader, etc., etc. And that's the conventional academia that I think everyone's aware of. But there are also alternative pathways in scientific careers. Industry is like the second more, it sort of goes academia or industry. They are the big two options. With industry, I have a lot of friends who are in biopharma companies here, doing downstream processing, bioprocessing, and food tech is also huge. You might be wondering, well, Mehershad, I'm doing life sciences. Can I still go into food tech? Yeah, you can. You actually can. A lot of the roles in biopharma, again, you don't have to be a pharmacy student to go into biopharma. A lot of the people that I know who are at these roles have done degrees that are very similar to mine. They have either done a molecular biology themselves, which is what I did, which is very close to life sciences anyway, or they have done a biotech, which is also something that you can do as a master's after your life sciences. You're still eligible for it. But it's also important to mention that most of these companies have different roles. So if you're interested in microbiology, you can still join food tech. If you are interested in processing, you can still, there are particular roles that you can go for these companies, which is really nice, because that means that it doesn't have to be, it doesn't filter you through just one degree. It's not that you have to have a food tech degree. There are a lot of roles, and a lot of roles require different skills. Then you can go to clinical research and pathology, which is also huge here in Queensland. It's also unfortunately huge now because of COVID. But these are also fields where I know people who have gone in these fields. Even without a master's, a bachelor's is sufficient. But I'll come to that later because there's an asterisk there. Then you can also go into teaching, as you may be aware. A few of my colleagues are actually in recruitment. So essentially they recruit some of the big people. They recruit for people who work in Thermo Fisher, for example. There's law, there's patent law, there's a lot of stuff with science now, especially now because in the last 10 years, I think there have been a lot of scientific companies and bio companies that have started. There's also business, which includes sales as well. A few PhD students from an institute next to mine that I know of, they've actually gone to work for Thermo Fisher, for example. If you're interested in science, but you don't necessarily want to have wet lab, if that's not your interest, but you still like science, there are these alternate pathways, even at really good companies. And then there's admin stuff as well. One of the things that I haven't mentioned here is communication. For example, the institute that I'm at, the Queensland Brain Institute, it has a communications department because, as I mentioned earlier, there's this push in the scientific community to want to reach the public, the layman, which means that a lot of these institutions, universities, they have these PR communication departments, but they're all pushing out scientific stuff. Maybe that's, sorry, I'll be very clear. I'll be very succinct. For example, the research that I'm doing. Now, if we publish a paper, the communication department from my institute would be like, hey, we noticed you published a paper. Now you published a paper to reach different scientists in your field. That's great. But we also want to reach the normal public. We want to reach students at the university who are not in science. And we want to essentially reach the general public. So they'll actually work with you to sort of put the importance of your research and the conclusions of your research in an article that is easily accessible. And then that gets pushed out in the public. A lot of the communication departments at the university also have tie-ups with journalists. So they'll get you sort of an interview at a radio station, or in many cases, people have gotten interviews with a journalist at a newspaper, or you'll be asked to come present your stuff at a TV, at a sort of this news science segment that happens on TV. So there's actually a lot of, and these are the things that I never knew about earlier. This just goes to show that as years go on, there are these things that there are these careers that you never thought were possible. One of the postdocs in my lab, actually, he was really interested in science, but he was also interested in graphic designing. So my boss, who's a very kind man, he sort of encouraged him to pursue graphic designing. Even though he was an exceptional scientist and my boss would have loved to have him in the lab, but he asked him to pursue his business and his passion. And he actually now designs a lot of the journal covers for the papers that we publish. So every time we publish a paper in a good journal, he actually designs all the journal covers, and then we send that journal cover along with our article. And then in a lot of cases, one of our journal covers that he designed has been on Nature Communications as well, which is great. Again, this is a career that I would have never thought of. So there are a lot of these opportunities, and I'll come back to this later. But it's not as conventional as you might think. And I'm sure there are a lot of things that I'm missing myself. I'll quickly go on to qualifications. If you want to have a scientific career, what qualification do you need? PhD is the most obvious and the most common one. I'll come back to how to apply to PhD. I just wanted to give a few tips. Masters is another big one. And honors, like I mentioned earlier, a lot of the technician, a lot of the pathology, clinical research positions are actually eligible. You are eligible for them even with the bachelor's. The reason I've mentioned honors is because it's really hard to get to find a job in clinical research if you don't have hands-on clinical experience, which we might not, which is why a lot of people that I know actually do an honors project, which is a one-year research project in a clinical research lab. So essentially, they work in a clinical research lab, they get all the clinical research skills, they acquire them, submit their thesis, pass the honors year, and then they go to the companies and they be like, hey, these skills that you're after, I have them because I worked at this research lab that does exactly the same thing. And then they get hired, and it pays really well as well. It's a really good role. Masters is the obvious one. That is what I'm doing. And then the PhD is obviously the best thing you can do. Now, PhD gets tricky because it's sometimes hard to apply. But one of the things that people normally stumble upon is if you're, for example, if you've not had particular research experience in a particular niche, and that's the lab that what you want to join wants to work on. People feel intimidated. They feel that, for example, I felt intimidated that I only know plant genetics. How do I go into neurobiology? But one of the eye-opening things that I've noticed is that people don't care. Most researchers, they don't care what your background is. They want to see if you're a good scientist, because you can acquire skills, you can acquire microscopy skills, you can acquire wet lab skills, you can acquire a particular technique. It's easy to learn, and that's not the barrier that we think it is. So all you really need to know, and I'll talk about this more later if you have questions about this. But very briefly, if you want to do a PhD, all you need to find is a lab that you want to work in, and an email of the person who's heading the lab, and you just need to send them an email, read their papers and tell them what interests you. And that's enough. I've known PhD applicants who have gotten into really good labs without any significant prior experience. Because what they really want to know at a PhD level is if you're interested in, if you're fascinated by the research, and if you're motivated to conduct the research. Everything else you'll be taught in the PhD. That's why you're still a student when you're doing your PhD. You're still learning. So you don't really need to know everything, even before applying. What are the things that you can do to help your CV? This is a big one. Again, things have changed now, especially in the last few years, because a lot of things are accessible online. Given that now you can access courses from a lot of the top universities, and you can learn a lot of programs online. Most programs that I've come across in science, in biology, have a free guide, a free manual, and tons of resources, everything for free. So the accessibility of these things has improved. All you need to do is you need to be motivated enough to learn a particular software program, or to undergo a particular online course. If you have time and if you're motivated, these are the things that I would highly recommend you would do, is because Say, for example, you want to join a lab or you want to join a particular role. Let's forget it's a lab, say it's a company. And if they're heavily using a particular software and you know an above average amount on that software, that's going to significantly improve your hiring chances. Because if you know the tools that they're working with, that's all they need really. And these things are available extremely easily online. So yeah, these are some of the opportunities that are available, the different qualifications at which you can do different things, and some of the things that you can do even now to set yourself up for your future. So these are future directions. I'm sorry if this is very boring. Let's move on to some interesting stuff. Okay, this is my last slide. I won't bore you any much. But these are some of the conclusions. These are just some observations that I have, and these are essentially, this slide is a slide that if I could send something in the past for myself, this is the slide that I would send. So I hope it helps you. Again, like I mentioned, there are a lot of caveats. Maybe they work for me. Maybe they won't work for you. Maybe they work for someone who's interested in a particular career. Maybe it won't work for someone who's interested in a different career. But it doesn't help. It doesn't hurt to just share these with you, just so you're caught up with the thought process that I have and the things that I would tell myself 5 years earlier. Again, sticking to the scientific research theme, phenotype versus genotype. What's more important? It's a trick question. Both are important. But essentially, this goes to say that a lot of people, when they're doing either their master's or their PhD, they're extremely, they're confused whether to look for the university first, or look for the lab first, look for the group leader first, or look for the field first. In my experience, and it seems that holistically in the experience of most PhD students who have completed their studies, in their view, the number one thing that you should start with is actually not the university, it's the PI, it's the group leader. The group leader and the lab culture, according to the PhD students I know, is just the most important factor that you should look out for. Now, I know it seems tempting to go for a very good university because going to a good university comes with status, and it comes with credibility and things like that. But in science, it doesn't matter. Nature, Science, Cell editors rarely look at the university that a paper is being submitted from. They look at the research. And also in my experience, even in my field, there are great papers, great publications coming from universities that I've never heard about. But good scientists are everywhere. It's possible that the bigger universities have more resources, and it's possible that they have more good scientists. But it's not the only metric that you should look out for. So look out for a group leader or a supervisor that you really go along with, because that will determine the success of your PhD more than anything else. The other thing I also want to mention is that over the years, we've had a lot of people apply to our labs from different universities after doing their PhD. They want to apply to our lab for a postdoc career. And I've been through a couple of these recruitment processes, and I've noticed that my lab and a lot of the labs, they're actually never concerned with what the actual topic was. There are people who have worked with mice who apply to our lab, people who have worked with E. coli that apply to our lab. Our lab focuses on C. elegans, but it's never a factor. The other thing that is never a factor is that what lab you came from really is not a factor. If your supervisor says good things about you, you'll be hired. What research you did is actually the most important factor, but it doesn't mean that your research has to be published at a top journal. It always helps if it does, but it's not the only factor. During the recruitment processes, I've noticed that people are normally counterintuitively more interested in what their process was, in thinking, and what their logic was in doing experiments. Because scientists at a top level understand that there's a lot of luck. Discoveries come with a lot of luck. But if you have a good process, if you have scientific rigor, if you're asking the right questions, even if you didn't get the answer in your PhD, if you're asking the right question, then that's the scientist that they want to hire, because they want someone to ask those questions and then get the right answer in their lab. So just understand that that things are a bit gray. You don't always have to go for the top universities. Just go for the right lab and the right supervisor, especially in your PhD. In vivo to in vitro, as I've mentioned, there are different stages of a scientific career. It can start from a bachelor's degree, an honors degree, a master's, a PhD, and it can go all the way to a group leader or a CEO of a bioprocessing firm. Now you might think, Mehershad, you went there for a master's, and maybe that's what everyone should do. I actually don't agree with that. Depending on your career, depending on your personal inclinations, depending on your circumstances, you can sort of you can switch streams, fields, and countries whenever you want at different stages. And I mentioned this point because I've been lucky enough to see people at different points join really good labs. For example, one of the PhD students that has recently joined my lab actually did her master's from India, from Noida. And she's brilliant, and she was very easily hired, not a question. So if you choose to do your master's in India, that is absolutely not a hurdle of any kind. If you do good research, that's all what labs are looking for. I say PhD because I know a lot of postdocs and group leaders and assistant professors who actually did their PhD from India as well. So just goes to show that the move between either fields or countries doesn't have to be at a particular point in your academic career. It can happen at any point. I also say group leader because the plant genetic stuff that I did in the Queensland, in coffee, the Queensland Alliance for Agriculture and Food Innovation. And the head of that department and that lab is Professor Nina Mitter, who's a huge name in the entire building and the entire university, actually the entire state. A lot of the politicians, a lot of senators, they actually come to meet her because of how influential her research is. And she did a PhD and postdoc in India, but she was so exceptional that that institutions in Australia, in the US, were just extremely desperate to have her join their team and bring her expertise. So there's absolutely no right way, or there's no particular point at which you need to sort of make a move again across countries or across fields. Obviously, I'm in Australia. I've not done research projects in India after JHC, so I don't know for a fact about the researchers in India, so I can't mention. But it goes without saying that you don't have to go outside India at all. There are brilliant researchers in India at institutions in India. I only mention these because these are the people I know firsthand. Plagiarism. Plagiarism is a big thing in science, but it's not that's not the topic that I want to talk about. By plagiarism, I mean sometimes it's easy to copy what other people are doing. Surely I was tempted. I almost took engineering. People who are doing engineering, I took it. Thankfully, the Bachelor's of Science admission in JHC happens before engineering does its admission. So I knew that I wanted to stay here. I know a lot of people do what they see other people do, and I don't necessarily hate that. I understand why people do it. On one hand, it's stability. If you want to do, for example, when I moved to do my master's, I had no idea. I had no one that I knew who did this in my circle. I just figured things by myself, and it was very challenging. There was no stability for a while, but after a point, you're very satisfied at what you're doing, because whatever you're doing is exactly what you wanted to do, because those are the decisions that you took. If you sort of go for doing things that are popular, that other people are doing, there's safety in numbers, but perhaps you will end up doing things that you necessarily are not 100% passionate about, maybe just 80% passionate about, and that's okay. You just have to choose which way you have to go, and I don't hate either of the ways. Novel discoveries in science, there are novel discoveries all the time. C. elegans, the organism that I'm working on, it wasn't an organism that people knew about 30 years ago. The entire field of neurobiology that stands that my lab focuses on didn't exist 30 years ago, which means that there are careers also that don't exist now that might exist in a while. So it comes again, you need to go for risk appetite versus passion. One example is, if I took all the career decisions in my life, aiming to be a scientific writer, maybe 10 years ago it might look silly, but now there's a lot of people want books about vaccines. So it exists. The career exists now. It's just a hypothetical example, and not a very good one. But what I'm trying to say is that it might seem that your passions don't align perfectly into a career. But I recently went to a conference in Sydney, and Martin Shalfie, the scientist who came up with GFP, green fluorescent protein, an excellent discovery in science, he was sort of asked a similar question about his research. And I won't focus on the question, but his answer was that either you can do things that make sense to an editor of a journal, or you can do things that start a new field. And his discovery, the green fluorescent protein, indeed started a new field in science. So again, you want to do things that align with your passion. And if a career doesn't exist now, that means that you can make that a career. You can be the start of that career. I understand what I'm saying is extremely risky. It sounds risky. So if you don't have the appetite for risk, it's fine. It's fine. But I'm just letting you know, if you're extremely passionate and a career doesn't exist now, it's possible it might exist. Recombination. In science and biology, there's obviously recombination that happens all the time. In my case, I found that the two things that I never thought, writing and drama and filmmaking and then science, I truly never thought that they would ever go together. I always thought they would be two things that I would sort of pursue independently. But sometimes there are overlaps. I'm interested in writing and journalism, so I have been contacted by a few news organizations to write a weekly column about science, about all the discoveries happening in science. The book obviously is a good example. If you're interested in science and you're interested in writing, write about science. That's easy. One of my dream careers actually doesn't exist probably, but I'm hoping it will by the time I need to pursue it. I like filmmaking, like I mentioned, and I like science, and a show about science would be great. Sort of think Art Attack that came on Pogo, but that was for art. Something like that for science, but for adults. That would be pretty interesting. I think a lot of people are interested in science non-academically. A lot of people want to hear about science, even as laymen. So yeah, there's a lot of recombination that happens in biology and in careers. So if you have two passions that you don't think will ever go together, maybe they will. And that's all I'm saying. Second last point of the entire talk, and I'm so sorry for going this long, is the lack operon. An operon is a cluster of genes that work together. And science is a team sport. In science, people often work together. People work together within a lab. Labs work together on a collaboration. People work together in a company, and companies also work together. So some of the things that people often overlook in a rush for learning how the PCR works or learning bioinformatic tools is that you still need soft skills, and you still need to be a kind of person who's a team player and is open for collaborations. A lot of the things that I see around this institute can be summarized by saying that people don't necessarily want to work with the best scientist. They are more happy. They're happier to work with the second best scientist who's a nice person to work with, if that makes sense. So even if you're not the most exceptional scientist, but if you're a good team player, then you're actually more of an asset to your lab, your company, your research institute, than being the absolute best scientist. Doesn't hurt to be the best scientist and a good team player. So aim for that, but don't forget soft skills and being open to collaboration. These are things that you will be hired on the basis of. Lastly, and this is the point that I'm actually this entire talk is just so I could say this, because this is something that if I could say anything to my past self, this is the only thing I would tell myself, is that I have one regret with my whole journey in life sciences. And I think this is a good point where I can tell you to not make that mistake. And that is, so there are exams that happen every year, and we study for the exams. And I know this sounds extremely cliche, but I genuinely mean it, is that I was really good. I got good marks, so I thought that I was really good at science because the marks were on a science examination. It turns out I was not really good at science. I was really good at exams. And I think, unfortunately, the schooling that I had and the colleges that I joined, they made me really good at giving exams. I'm really good at topping exams, but I'm not good at science because I never learned how to do science. I just learned how to give exams. You get a question bank, you learn the answers in the question bank. But the thing is that, and I know Jai Hind is now autonomous, but back then you could omit certain chapters, you could omit certain topics. And neurobiology is a topic that I omitted. I got great marks. I never knew neurobiology. Now I'm working in a neurobiology institute, and that's horrible. What it means is that I have to work so much harder, and I feel that if I had to work hard now at this point, I could have just worked harder back then, instead of going the other way around, instead of going the long way. So I feel like I know exams are extremely deterministic. I know exams and the marks, they all mean a lot, unfortunately. But just try to understand the science. The one thing I want to mention is that and I think Srilata ma'am would remember this, is that for immunology we had this book that we called QB because that's the author's name, and I never studied immunology before the exam, and I never studied genetics, which Nilufar ma'am taught before the exam. I just attended lectures, but with full concentration. This is not even a joke, and I genuinely never studied before the exam. I wrote all the exams only on the basis of what I remembered from the lectures. So it turns out that if you want to have a great scientific career, it's extremely easy. All you need to do is be focused during those lectures. And I know it sounds boring, and I know it sounds cliched, and I understand if someone said this to me 5 years ago, I would laugh at them. I get it. But I have to say this because it would extremely go a long way in helping you. So just focus on the science. Don't focus completely on question banks and exams and answers that you need to mug up, and focus in lectures. That's genuinely where all my learning happened. It never happened after. It never happened the night before the exam. So I would even go as far as to say that if I had paid more attention to the science, maybe I had a Nobel Prize right now. I'll say it. So if you want a Nobel Prize, pay attention. And I know it sounds boring coming from me, but genuinely do. So that's all I had to say. I'm happy to cover any specific questions that you have. It's obviously hard to cater to everyone's individual careers and interests in a common slide. But this is the gist of it, and I'm very excited and interested in knowing what questions you have. Sorry for taking the time, and thank you for having me.
Thank you so much. Am I audible?
I think you'll have to mute the computer that's showing the camera. Was it muted?
Hello.
Yes.
Hello. Hello.
Yeah. This is great.
Thank you so much, sir, for this very insightful and interesting presentation. I personally thought that it was very unique of you to, club those explaining us using the metaphors. So that was very interesting.
Thank you.
We
Thank you.
have a few questions for you.
Yes. Yes.
Thank you. Hello, sir. Firstly, thank you so much for this wonderful session. Some of us have a few questions from you. With the first question going ahead, like you said that any time of your journey, you can change your fields or you can just change your trajectory. For me personally, I am very much into neuroscience, like from school. So I am very sure that if I want to do something, it's neuroscience. So for someone like me who is very sure, what would you recommend for masters, like a broader spectrum or concise myself with neuroscience?
I think am I audible?
Yes, sir.
I think first of all, it's brilliant that you know exactly what you want. I think that's very rare, that having that clarity. So already congratulations on that. I think if you know the field that you're interested in, then you can specialize further. I think being in life sciences helps give you the basic information on most of the things that you need to know. So even within neuroscience, it might be possible that the project that you're working on involves, for example, I'll just give my example. We work in neuroscience, but we work using the microscopic worm C. elegans. C. elegans eats its bacteria. So you need to know a little bit of microbiology for some of the experiments. But it's not the level of microbiology that you need to know that you would have to do a master's in microbiology. I think life sciences gives a great platform where you have a holistic understanding of all the fields, and now you can go further. So I think if you like neuroscience, if you're passionate about neuroscience, I think you just go and specialize. I think that is how I would do it. Yeah.
Thank you so much, sir. The other question is that you were talking about some basic skills that are required. So there might be some basic that would be, a mandatory kind of thing for all the labs all over the world. So can you share some of them with us?
By basic skills, do you mean on the soft skill side or on the technical skill side?
Both.
Soft skills, I think you need to be able to communicate. Science is all about doing science and communicating science. So communicating your science, being passionate about communicating it, being clear, in being able to get other people interested, because obviously you're excited about your science, but you need to make sure you get other people excited as well. One of the very crucial and fundamental things about science and academia is grant writing. And grant writing is essentially convincing people that what you're working on deserves attention, deserves further research. So that goes a long way, not only as a master's or a PhD student, but also when you're a group leader. And you need to convince greater funding bodies to let you continue your research. So soft skills would include communication, working in a team, collaborating. I think in one of the hiring interviews that I had, the professor was taking the interview, asked if I liked being alone or liked being with a group. And I said both. And he said that's the right answer, because science involves working alone a lot of the times, but also collaborating the other times. So you need to sort of have the switch where you're okay with the menial day-to-day tedious work of just working by yourself on your project, but you also need to collaborate when it requires. So that's on the soft skill side. On the technical skills side, this might sound very basic, but Excel is actually, for some reason, the scientific community is still hung up on Excel. People still use Excel all the time. I think most labs that involve molecular biology would require you to understand a PCR, but not to do it necessarily, but just understanding so that you can troubleshoot it when it doesn't work later. And most PCRs don't work. At some point, they will not work. This is just a common gospel in science. But if you understand how it works, then you know how to troubleshoot it. So just general troubleshooting and logic. The other thing is general understanding of a microscope, so that you can troubleshoot later. And I would say lastly, technical skills would include, I guess it would also include writing, because even if you're a master's student, you have to write a thesis. PhD students, you have to write a thesis. Group leader, write a grant. So writing science, communicating science, understanding of basic skills like PCR, microscopy, etc. But if there's a particular field, then because then there's a lot of things that are very field specific. So yeah.
Hello, sir. So you mentioned that you skipped neurobiology in your exams, and now you are studying it. So my first question is, what made you switch from plant genetics to neurobiology? And my second question is that did you ever know or even think that during your BSc this day would come or this decision would be made?
It's a great question. First of all, I'll start with why I switched from plant genetics to neuroscience. The answer is very simple: is that I didn't actually switch. I'm interested in genetics from bachelor's. Like I mentioned, I enjoyed Nilofer ma'am's on lectures on genetics, and I was, I think, more inclined towards genetics, and then in immunology, and then environment science, and then neurobiology. So the genetics is what's interesting. So it was genetics in the plant. It doesn't matter what the organism is. It was Arabidopsis, which is the plant model organism, and now it's genetics in the worm, in the nematode model organism. So the alluring part about these projects is still the genetics for me. Unfortunately, things in science are not. You need to all projects look at things holistically. You need to consider so many things. So you just can't just look at genetics. You have to understand the neurobiology to understand the genetics behind it. So the switch was because I just like genetics. Unfortunately, there's other things that comes with it. So earlier I had to learn about plants, and I didn't enjoy botany as much. And now I have to learn neurobiology. And so far, the way things are going, I'm sure I will have to learn the environment sciences next, because all the fields that I ignored during my bachelor's, they seem to be coming back in my career. I think the second question was, sorry, I completely forgot the second question.
So the second question was, did you ever think that you would make this decision while you were in BSc? Did you know any of these things would happen?
No, absolutely not. You saw the photos. That person didn't know anything. I didn't know I would do a master's. I thought I would do a master's, but I didn't know I would do it. I never had plans of going to Australia or any other country, actually. Yeah. So sometimes things are planned, which is why I really admire people who can plan things and who have clarity, because I didn't have those. I just sort of went with what felt right.
So I'm pretty sure most of the students here relate. Most of us are just going by a day, making decisions in the moment. So the next question is that you mentioned you want to come up with a show about science, something like Art Attack, but science-based. Now, say if that day did come, and we hope it does, would you be ready to, stay out of the lab and not really be working on science and just be concentrating on that, or would you still like to pursue both simultaneously?
Again, this is a great question, by the way. I just need to take a moment and say that these are great questions. Thank you so much for asking. Thank you for valuing this talk. And just understand that if I was in your place, I wouldn't ask any questions. I would have ignored the talk. I would hate it. It would be a waste of my time. So thank you for being better than I was. This is a great question because thankfully now I have people in popular culture who are doing these things. I don't think necessarily there's a large scale science show that exists, but there are a lot of researchers. There is actually a neuroscience researcher from Stanford who has a podcast, and he's so successful that now his podcast actually pays him more than his science. And Stanford is getting so much traction now. I mentioned Stanford, right, because of the podcast. So it's getting so much traction because of this podcast that Stanford sort of like, please keep doing the podcast. Even if you're not doing research, just keep doing the podcast because it helps us so much in publicity. But he still has a lab. He doesn't. So he's a group leader at this point. So he sort of automated some of the things to the postdocs. He doesn't get so actively involved anymore, but he still has a lab. And I think that's the way to go, is that you don't want to lose touch. you can sort of do it on a part-time, like you can do it 50-50. I think that's the way to go. I think if you just completely remove yourself, science advances so fast that that you'll just be lost. You'll not know what's really actively happening in the scene. And that's what the people want you to communicate. People don't want you to communicate what's there in the textbooks because that's already been printed. People want you to communicate as an active researcher what's happening now. So I think you have to be in the know-how, even if it's partially.
Thank you so much for answering those questions, sir. This was definitely fun, and we all hope you get your Nobel Prize that you deserve to. Thank you.
Thank you, thank you, thank you. I'll just put it out there because I used to be shy and embarrassed. So I'll just say that you saw embarrassing photos of me. So there's no reason to be embarrassed. If you have any last questions, just ask away because you know, it's fine if you embarrass yourself.
Anyone, any questions?
Sir, I think we don't have any questions.
That's fine.
No, ma'am, there's something wrong with the PPT. One second. There is something wrong. Okay. Now I would like to call upon Sarah for giving the vote of thanks.
Hello, sir. And hello, everyone present here. I'd like to take this opportunity to thank Mehershad sir for taking some time out from his busy day and having this informative and fun session with us. I'm sure each and every one of us had a great time, and we got to learn something valuable from today's talk. It was an absolute honor and delight to hear about your impeccable journey and all your achievements. And personally, so you're very, very inspiring. I'm truly inspired by you. And
Thank you, thank you.
yes, it was amazing. And yet you're very relatable, humble, and fun. It was just fun. Yeah, it was actually very fun. Thank you so much, sir, and thank you so much for answering all the questions so patiently. On the behalf of the entire Life Science, I'd like to again thank you so much. Thank you for
Thank you.
with us today. And yeah, thank you.
Thank you, everyone. Thank you. Thank you.
Thank you so much, sir, again. And I would now like to call upon Nilofa ma'am to say a few words.
There's nothing to say. All that I'm
It's-
glad that
I mean,
at least,
yeah.
you're doing life sciences. I mean, you're doing genetics. At least someone is following what I taught, man. Thanks so much, Mary.
No, anytime. This is very, very helpful for me as well. Because it really, I mean, in the day-to-day, you don't notice what you're doing. You don't see things from a broader perspective, but this forces me to sort of look at the entire journey, and this makes me really, really glad. So thank you so much.
India.
December, Jan. Oh, hopefully next time in person.
Yeah,
The
next time in
last one
person.
was in person.
Yeah. And by that time, you should have 2-3 more books published, you know. Then
I'll
we'll
try,
set
I'll
up a
try.
table for you here and everyone will come and take your autograph and all. And then I'll sell it for $5,000 or something. Anyway, thank you so much, Mary.
Thank you.
Bye.
See ya.
Bye-bye.
See ya.
Say bye. Say bye. Everyone is saying bye to you.
Yeah, no, I see. Thank you, everyone. Thank you, Zenia. Thank you. Hi, Sakina.
Now wait. We'll stop the YouTube streaming and you all can sit and talk. Wait. You all can
Just a minute, sir.
Just stop the streaming.
Yeah.
End of the streaming is you coming.
Okay, done.